The second to last talk of this morning session given by Juan Sebastian Vargas Soto.
So this is a very brief summary of the work that I did for my PhD. I'm currently a postdoc with Peter Molnar at the University of Toronto, but I will be looking for further employment at the end of this year. So if any of what I'm about to talk about catches your eye, please come and buy me a coffee. So following up on the previous talk, the main argument that I want to make today is that finding or identifying these spillover events is very challenging, no surprise. And in particular, when we're dealing with wildlife species and rare, which are also commonly endangered species. We know in general, like where these events are likely to happen. This is a map that shows the risk of zoonotic events worldwide. But some of this also applies for the risk of transmission between wildlife and domestic species. And we see that the highest risk are in areas where there are many different species, especially if they are related along the same order or genus. So if we're talking about mammals, if there are a lot of mammals, that increases the risk. But also where there is intensive land use change. And where these conditions kind of compound is in many tropical regions. And I will focus on one region in particular, which is here in Central America, for many reasons, none in particular. The reason that I wanted to escape the Canadian winter to go do fieldwork. So this is the Osa Peninsula in southern Costa Rica. It is a region that is well known for its biodiversity, and that's largely due to conservation efforts, the establishment of protected areas in the 1970s, and then more recently, the establishment of programs for payment for ecosystem services. And they have managed to increase and sustain forest cover of upwards of 70%. And it is home to now a number of, or a high biodiversity, including many charismatic and species of conservation interest, like jaguars, white-lipped peccaries, and tapirs, all of which are endangered or threatened to some degree across their global ranges. So I'd like to use this system as a case study and ask one overarching question. It's very simple, or it seems very simple. What is the risk of spillover between wildlife and domestic species? And the way I've organized this is I've approached it from three main directions, trying to find out things about the host ecology, about the parasite ecology, and then using field collected data, and then integrating this into a quantitative approach along with data from the literature. So first, we need to understand very basically what are the drivers of the distribution of the different species that are in this area, and based on that, which species could be most at risk for there to be spillover events. And this is work that I conducted in collaboration with a number of different institutions, both in academia, both local universities and international universities. Well, important. Government institutions, non-government organizations, but also, and very importantly, local businesses and communities that help out with the sampling. And this work that is published in Conservation Biology. So what we did is we went out and put out a lot of camera traps wherever we could in a sort of a organized grid, both inside protected areas, but also outside protected areas to get this kind of gradient, just one of the variables that we were interested in. Then we analyzed the occurrence data using a joint species distribution model, which is a type of occupancy model, to try and link the observed pattern of detection with some environmental variables of interest and try to see what the effects of those environmental variables were. And we used four main covariates to represent the general level of anthropogenic disturbance, the habitat quality and availability around the different stations, and also whether stations were protected or not. So what did we find? Out of these four covariates, the general level of disturbance had the highest effect on most species, or the most significant effects, but the effect varied across species. You have, for example, some that respond negatively to anthropogenic disturbance, meaning that they will be found more likely in well-forested areas away from human settlements, like the jaguar peccary, large species that are specialists. But you have also the opposite with opossums and armadillos, so more generalist or insectivore species. They are more likely to be found near human settlements. I will actually not talk about any of these species. I will actually talk about the ocelot, which is an interesting case because it didn't respond either negatively or positively to anthropogenic disturbance, meaning they are widespread throughout the region and they'll be found both in the forest, but they'll be also coming into contact with human settlements and the domestic species associated. So now that we have some information about where each species are, we can ask questions about host abundance, trying to dig deeper. And host abundance, as you all know, is a key determinant for epidemiological dynamics. And for this, I again collaborated with local NGOs, and this work that is currently being reviewed in biodiversity and conservation. Again, we went out and put a bunch of camera traps in different grids along 3 years. And then we identified ocelots individually based on their unique spot patterns. So this is a very brief example. The animal on the left is the same as the one on the bottom right, but not the same one as the one on the top right, clearly. And then, because we have individual identification, we can use spatial capture-recapture framework to estimate the density in our study region. And we estimated a density of 34 individuals per 100 square kilometers, which is among the highest reported using this method. And that corresponds to roughly 300 in the whole peninsula. Something that we found that was very interesting is that the ocelots like to use man-made trails and roads a lot. And that puts them at risk because, as you see, these are the same trails that dogs are using during the day, and that puts them at risk of spatial overlap, if not temporal, that could lead to the spillover, particularly of environmentally transmitted parasites. If you're still playing, this is also not the same individual. So now that we have some information about the host, we can then focus on the parasites. And as I said, we were focused on parasites of environmental transmission of dogs and ocelot. But we wanted to identify everything that we could find and estimate the prevalence of some particular parasites of interest. So for this, I went out by myself and with some trained scat detecting dog all along the region, inside protected areas, outside in towns, and collecting scat for wild animals, for ocelots and pumas as well, but also domestic dogs. And using microscopic analysis, we found some of the common parasites that you would expect. And the ones that were most prevalent were this group here, which are strongylobed worms, hookworms. Well, we presumed they were ankylostoma at the time. We had to confirm that, both in the domestic species and in the wild species. And it had prevalence upwards of 70% in the dogs, which is not surprising. This is one of the most common endoparasites of dogs worldwide. But the prevalence in the wild animals was also relatively high. The problem with these species is that all the worms or the eggs of these worms within the genus look exactly the same. So we need molecular methods to actually confirm that it was the same species in the wild and the domestic animals. So we conducted PCR and DNA sequencing, and we found that, unsurprisingly, the species that we found in dogs is Ankylostoma caninum, the common dog hookworm, although we also found another species, the Ankylostoma ceylanicum, which could be of interest because it is more zoonotic, it can actually cause pathology in humans. As for the wild felines, well, we could not identify the parasite, and this is most likely due to degraded DNA in scat that is out exposed to radiation and desiccation in the field for some long time. We can, nonetheless, with the information that we have about the host, about the parasite in the domestic species, and with information from the literature, integrate this into a model and try to determine what is the likelihood of spillover. Even if we can't confirm it, what is the likelihood that it is occurring, and does it fit the data that we have? And then potentially ask about some also alternative mechanisms that could explain the prevalence. So we built an epidemiological model. This is a 2-species model that are sharing an environmental pool of infectious stages. This is inspired by some of Andy Fenton's work. And based on this, we are focusing on the prevalence, the expected prevalence, and we're focusing on 2 main free parameters, if you will, which are the level of overlap between the two types of hosts, shown here on the x-axis, and the level of compatibility of the parasite, which we presume is typical of dogs, the level of compatibility with the feline species. If you remember, and this is focused on ocelots, we found a prevalence of 20% roughly in ocelots. And as you see, if there is high overlap and high compatibility, you would expect high prevalence, not surprisingly. But however, if either of those is low, then the expected prevalence is also very low. And the data that we have is actually a very low compatibility between the wild feline and the parasite, and the expected prevalence would be less than 5%, so much lower than what we found. So what is an alternative mechanism? Well, that there is, even if there was higher overlap, which could be driven by land use change or higher abundance of dogs, the prevalence will still not go over 10%. An alternative mechanism is that there are two separate parasites in the wild and the domestic species. And in that case, the compatibility would be much higher. And even with low habitat overlap, you would get the prevalences that we observed. So we still have to confirm that, whether it is a different parasite, but that is looking like the most likely explanation. So coming back to my main point, like what is the risk? The main point is actually that this requires a lot of work, and some of the challenges have already been mentioned in the session today, or some of the solutions is interdisciplinary work, and also something that Jessica brought up in the case of the chimps, and something that I'd like to highlight as well is the collaboration, and more importantly, the engagement and skill building in the local communities as a tool to monitor and identify not only the threats, but also to monitor in near real time the threats for wildlife species, some of which are endangered, but also obviously for parasites of human interest. With that, I'd like to thank everyone that helped out with the thesis, the people that actually started this work more than 10 years ago, and obviously the funders and all of you. And I'll be happy to take any questions. Thank you.
Thanks. We've got time for one or two questions.
Hi. We've been doing some work with climate change modeling, looking at whether we can use model climate change to predict disease risks. And one of the things we've been struggling to find, but I know they're out there, and this appeal both to you and to the entire room, is robust but very granular epidemiological data on diseases that are known to be climate affected, and particularly affected by precipitation and humidity, we're looking at specifically. So it's really a question, rather than about your presentation, of have you got data sets that would fit that we might be able to use and collaborate with you to run through climate change models? And if you don't have them, if anybody else has them, this is what I look like. Please come up and talk to me afterwards. And if anybody wants to go through, I've got some kind of the details of where we've got to with the research on my laptop that I can talk through with you. Thank you. Sorry.
So the question is about the availability of climate data in this specific region, just general. Yes, some depends on the type of data that we can talk some more.
Great, thank you very much.